Alright.
Hello everyone, I'm Liu Yang, a master student from Gamma Lab at Beijing University of Posts and Telecommunications. My advisor is Sichuan. It is a great honor to present our work, which has been accepted at SML 2024. And in this study, we mainly focus on improving the architecture generalization of graph distillation based on the spectral graph theory. Now, let's begin with the definition of graph distillation, which aims to distill the knowledge of this large-scale row graph into the small syzytic graph. So the input of the graph distillation consists of adjacency metric, node features, and the label, and the output is A prime, X prime, and Y prime, where the Y prime is usually predefined according to the class distribution of Y. Once we get the informative syzytic graph into the, we can use it to train the GEN instead of this large-scale row graph, thereby accelerating the training process and reduce the time and space overhead. And the pipeline of graph distillation consists of two steps, including of distillation step to get the syzytic graph, and the evaluation step to evaluate the genes trained on this syzytic graph. And it is noteworthy that both steps involves specific gene, and we refer to them as distillation gene and evaluation gene respectively, and which will be frequently mentioned in the following discussion. And the direct solution in distillation step is formulated as firstly obtaining a GN that is theta G prime by minimizing the training loss on this syzytic graph. And then this syzytic graph is used to minimizing the training loss on the row graph to optimize the syzytic graph. And because theta G prime depends on the syzytic graph, so the outer optimization incorporate the back propagating through the inner training, which leads to a bi-level optimization with high computational complexity. So a serious work has been proposed to approximate the process. Specifically, the large real graph and the syzytic graph are both fed into the same distillation gene to get the model-related information. And then the syzytic graph is optimized by minimizing the discrepancy. And there are three traditional methods of graph distillation. The first one is gradient matching, which optimizes the syzytic graph by minimizing the discrepancy between the gradients computed on the row and the syzytic graph. And the second one is trajectory matching, which firstly obtain the model parameters trained on the row and the synthetic graph. And then the synthetic graph is optimized by aligning these two sets of parameters. And the third one is distribution matching, which optimizes the synthetic graph by minimizing the discrepancy between the class embeddings of the row graphs and the synthetic graphs. And it is noteworthy that existing method all involves the specific gene. Therefore, a natural question arises whether the different distillation gene affects the synthetic graph and thus affect the performance of different evaluation genes. So our work begins with extensive investigation experiments, and we find two main limits. The first one is that existing method introduce distribution bias in the synthetic graph. In this experiment, we use a low pass gene and a high pass gene to generate the synthetic graph. And then we compare the total variation distribution with a row graph. And we observe that if we use a low pass gene, the total variation of the synthetic graph is smaller than the row graph. While using a high pass gene result in a higher total variation. So we claim that existing methods introduce different distribution bias with different distillation genes. And then we also evaluate the cross-architecture generalization of existing method. Specifically, we adopt 6 distillation genes to distill the row graph, and we can obtain 6 corresponding synthetic graphs here. And then we train each synthetic graph using this 6 different evaluation genes. So the D denotes the distillation genes and the E denotes the evaluation genes. From each row, we can observe the significant performance gap. And from each column, we can observe that the optimal performance is achieved on different synthetic graphs. That means if we want to obtain the optimal performance, we have to traverse the various distillation genes and select the optimal performance. It is not efficient. And an ideal solution is that we only need to perform the distillation once, and then we can use a single synthetic graph to train the various evaluation genes, and while achieving comparable performance. To realize this goal, we naturally ask what causes the performance discrepancy and how to reduce the significant gap. And then we provide a theoretical analysis of existing distillation, taking gradient matching as an example. Firstly, we adopt a unified formulation of genes, involves propagation operation followed by a linear transformation. So the objective of distillation genes can be simplified into a standard MSE loss. And based on the training loss, we further derive the gradient on both the row and the synthetic graphs. And the gradient matching aims to minimize the discrepancy between these two gradients. So we substitute the gradients into the MSE loss, and we further derive an upper bound of this gradient matching. And we can see that the upper bound consists of an unsupervised loss and a supervised loss related to the downstream task. And these two terms are the target distribution that the synthetic graphs should align. Now let's we focus on the unsupervised target distribution. And because the GL can be decomposed as U times diagonal metric G lambda, and then times the transportation of U. And this formulation can be further expressed as a weighted summation over the eigenbasis. So the unsupervised target distribution can be formulated as this equation. And the unified formulation of Gn can be also interpreted from the perspective of the spectral domain. Specifically, the first step is to transform the graph signal in the spatial domain from the spatial domain to the spectral domain, and we can get the magnitude at different frequencies. And then the G lambda acts as a decide filter to modulate the magnitudes. And then the filtered signal are transformed back to the spatial domain through the inverse operation. In this view, different distillation Gn corresponds to different filtering functions. And when the Gn stacks multi-layers, that is T increases, the matching loss tends to emphasize the terms where the G lambda I is greater than 1, because the corresponding weight G lambda I to the power of T is larger. So this analysis explains why different distillation genes leads to different distribution bias in the synthetic graph. And inspired by this analysis, we design a model agnostic graph distillation, and we only matches the environment information. So in our first matching objective, last year, namely our eigenbasis matching, we exclude the weighting effect of G lambda and treat the eigenbasis term equally during the matching. And because the eigenvector U is naturally normalized and orthogonal to each other, we introduce an additional regularization to further constrain the representation space. And in practice, we found that the eigenbasis matching improves the generalization but contribute less to the performance of node classification, because they neglect the information related to the downstream tasks. So inspired by the supervised loss of the upper bound, we introduce a discrimination constraint. Specifically, we first calculate the node embeddings and then obtain the class level embeddings by summarize the node embeddings within each class, and the loss D aims to enhance the intra-class similarity and uh, enforce the interclass orthogonality across the class embeddings. And it is noteworthy that the loss D introduce a slight bias because the calculation of the class embeddings incorporates the spectrum, and will introduce a low frequency preference. However, it is inevitable to get better performance, but we can adjust the weight of eigenbasis matching and the discrimination constraint to balance the generalization and the performance. So this is our overall orthogram, and this is our total loss. Yeah, it can be seen that the eigenbasis matching, the discrimination constraint, and the orthogonal constraint are adjusted by different weight. And by minimizing this loss, we can get the syntactic eigenvectors and the node features. However, the syntactic graph structure is implicit due to the absence of the spectrum. And ideally, if the syntactic graph is to preserve the structure information of the row graph, its spectrum should be similar to the row graph. So we directly replicate the spectrum of the row graph and combine it with the syntactic eigenvectors to constrain the adjacency metric of the syntactic graph. And this is a method of our GDEM. And extensive experiments demonstrate the effectiveness, efficiency, and the generalization of our method. We firstly evaluate effectiveness on node classification task. We observe that GDEM achieve state-of-the-art performance on 6 out of 7 data sets. The results reveal that our eigenbasis matching strategy can also yield good syntactic graphs without relying on the distillation gene as previous method do. And we notice that GDEM perform slightly worse on RCAF, and this might be because under very low compression ratios, the number of the available eigenvectors are too limited to cover all the important subspace, and its experimental verification can be found in our appendix.
Sorry, I have a question. Do you mind if I ask
Okay.
a question before you keep
Okay.
going? So just to make sure I understand, it sounds like the question is what size is the synthetic graph in relation to the full graph? So it sounds like that is what you mean by ratio, but I am not sure I understand.
Oh, sorry. The ratio means the the number of the synthetic graph. Uh, uh, yes,
Proportional to the size of the original graph.
yeah, yes,
Okay.
yes,
And then I have a
yes.
follow-up question. For some of these, for example, the value for the ratio is bigger than 1%. It's 100 and it's 1.220%. So basically that means that you take 1% of all the, or something like that, like the size, the number of nodes in the synthetic graph is like 1% of the nodes in the original graph. Did I understand that correctly?
Yeah, yes.
Okay, I understand. And then the evaluation here. So when you do the evaluation, are you evaluating on the Y bar or Y dash, or are you evaluating on Y? Maybe I did
Uh,
not understand, but as far as I understood, you have on the first slide you had that the large graph has right like A, X, and Y, and then on the synthetic graph you have A bar, X bar, Y bar.
oh, okay.
So the evaluation that is on X, the node classification between X bar and Y bar. That is the results here. Did I understand that correctly?
Um, they they U prime. Yeah, the U bar means the synthetic. Yeah, the U bar means the synthetic eigenvectors, and
Mm,
we directly replicate the spectrum, namely eigenvalues of the row graph, and to constrain the A bar. Yes, the calculation of A bar is shown here.
okay, I understand. Thank
Yeah,
you.
it means the graph structure of the synthetic graph.
Mhm. But it is not the same representation of the graph structure as a normal adjacency matrix. It's slightly different.
Yeah, the method to synthetic the graph structure is different with the baselines.
Gotcha. Okay, cool. Thanks.
Yeah, because the spectrum the property of spectrum can preserve some important information of the row graph. So we preserve the original spectrum, and then since we generate the U bar, yeah, and then we construct a new structure of the synthetic graph.
Got it. Thank you so much. Sorry for pausing you.
Okay. And then we evaluate the generalization ability of synthetic graph distilled by different methods on four data sets. Specifically, each synthetic graph is evaluated on these six evaluation genes and the average accuracy and the variance. Sorry, this means the variance, and it is a mistake. And the average accuracy and the variance are reported in the table. We observe that the GDEM achieves the highest average accuracy on 3 out of 4 data sets, and moreover, GDEM consistently achieve the lowest variance, suggesting more stable performance across these different evaluation genes and highlighting its better generalization. Yeah. And then we also compare the optimal performance of GDEM against the baselines. For GCount and SGDD, we traverse 6 distillation genes We select the optimal performance for every evaluation gene. And because the GDEM is model agnostic, so it synthesizes only a single graph. Then we evaluate the synthetic graph on this different evaluation genes. And even in this setting, we can see that GDEM outperform the baseline in 4 out of 6 cases. And we also report the time overhead for achieving the optimal performance. And we can see that GDEM shows significantly lower overhead, highlighting its inefficiency. Then we conduct some ablation studies on the GDEM. From the table, we can see that the lossy eigenbasis matching and the orthogonal constraint are crucial for the better generalization, because removing either leads to significant increase in the variance. And on the other hand, the loss D, that is discrimination constraint, slightly hurts the generalization. However, it improves the overall performance. And the hyperparameter analysis is showed in these figures. The green lines means the average accuracy, and the blue lines means variance. We can observe that as the alpha increase, the variance gradually decline, means indicating better generalization. And however, a larger value of alpha leads to a slight drop in the average accuracy. And on the other hand, increasing the beta, the variance gradually increase. And when the beta exceed a certain threshold, the accuracy will decline, because the poor generalization negatively affect the overall performance. And we also virtualize the total variation of the synthetic graphs distilled by different methods. And we can see that the total variation distribution of the synthetic graph generated by GDEM is the closest to that of the row graph. And we also virtualize the total variation at the different training epochs of GDEM. We can see that the total variation distribution gradually approaches the row graph, indicating continuous improvement in the distribution alignment. Yeah. And here is a brief conclusion of our work. That's all my presentation. Sincerely, thanks for your attention.